So thanks everyone for coming today. So this is hopefully one of our first installments of gene editing kind of educational session. And so this is sponsored by the Masonic Cancer Center and also the Genome Engineering Shared Resource, which we'll talk about our services later on at the end of the talk. And so the first half of the talk will be presented by me. I'm Branden Moriarity. I'm the co-director of the Shared Resource. And I'm going to give you about 30 to 40 minutes about background on genome engineering and where it's come over the last 20 to 30 years. A lot of this will focus on CRISPR-Cas9, as this is the genome editing tool of choice, these days at least. And then the second half of the talk will be presented by our coordinator of the Shared Resource, Ruis. And he will get into the nitty-gritty details of how to generate CRISPR reagents to generate knockout cell lines for basic research, and how to actually implement those and identify gene-edited clones for your research. And we'll have, I think, extra time for questions and answers, with the goal of everyone here leaving and being able to go back to their laboratory, design and implement their own CRISPR guide RNAs for gene editing of their cell lines of choice. And so here's an outline of a talk. I'm not going to go through it all now, but you can see we're going to cover a lot of material. And again, it's on the topics I just described. So the first thing I'm going to start with is the basics of genome engineering using targeted nucleases. So what are targeted nucleases? They're actually proteins that can identify a specific sequence of DNA and break the phosphodiester bonds of that DNA to induce what we call a double-stranded break. And so there have been a number of iterations of these that have evolved in nature and also have been made by humans for research purposes over the last 20 to 30 years. So in nature, one of the very first targeted nucleases that was identified was the meganuclease, shown up here. And so this actually has a very confined sequence specificity that has evolved over time to target a specific sequence of DNA. And so originally people did try and modify the specificity of these, but it was rather challenging. It was basically structural protein biology. But actually then, just in the last 10, 15 years or so, they developed zinc finger nucleases. So these are man-made nucleases where they actually took zinc finger DNA binding domains from proteins found in mammalian cells, and they fused them to a nuclease domain called FOC1. And this is a dimerizable nuclease. And so they were able to engineer these proteins to bind specific sequences of DNA juxtaposed to one another, and when they both bind, the FOC1 domains come together and induce a double-stranded break. And then a few years after that, there was actually a large discovery here at the U of M by Daniel Voitas, where they discovered a new DNA binding domain in a protein family called tails. And these are from bacteria that invade plants. And so they developed a similar system where you could engineer these proteins to bind specific sequences that were juxtaposed to each other, and then again using the FOC1 nucleases to induce a double-stranded break. And then more recently, I'm sure you've all heard of the CRISPR-Cas9 system. And so this is developed from the adaptive immune system in bacteria, where they actually have a constant protein. So you do not have to do protein engineering. And in this case, it finds its cognate DNA sequence using a guide RNA. And so what this is it's a constant backbone of RNA of about 80 base pairs, and then a specific sequence that targets the DNA for a double-strand break induction. And the only caveat here of targeting essentially any 20 base pair sequence you want is that it has to have this PAM sequence next to it, proto-adjacent motif. And that is typically 3 base pairs for the canonical Cas9, and I'll talk about some other Cas9s that have been identified as well. And so these are what targeted nucleases are, and kind of the different flavors of them that have been developed over the last 20 to 30 years. And so now we'll kind of focus in on their usefulness in the lab, and then specifically with CRISPR-Cas9. So why do we want to use these targeted nucleases in research? And so like I mentioned earlier, the specific task that is carried out by targeted nuclease is to induce a double-stranded break in cells. And so this double-stranded break is useful for research because of the way the cell repairs these double-strand breaks in DNA. So typically in mammalian cells, about 95% of the time when you make this double-stranded break, it's going to be repaired by a pathway called non-homologous end joining. This is a very error-prone pathway. It essentially just tries to sham the DNA ends back together, and you frequently get addition of nucleotides or removal of nucleotides, and these are called insertions or deletions or indels. So you'll hear this word indels a lot in genome engineering. And the reason this is useful is because if we get an indel formed here in a coding region of a gene, we can actually put that gene out of frame such that it can no longer make its protein, thereby knocking out the function of that gene. And so this will be the focus of Brian's portion of the talk, is how to design CRISPR-Cas9 reagents to knock out genes in mammalian cells. Now, the other really useful thing that we can do with this technology, or this double-strand break that we've induced, is it can be repaired in another pathway in the cell called homologous recombination. And this is typically done in the cell by using the sister chromatid to perfectly repair a double-stranded break that would occur just by chance in the cell. But we can actually take this opportunity to further manipulate the cell by providing a donor DNA or plasmid DNA to the cell that has homology to the region where we've induced this double-stranded break, that then we can actually insert heterologous DNA sequences to actually change the coding DNA of a cell. And so with this, we can change single nucleotides, single amino acids, or we can actually insert large cassettes like GFP or other tags that you want to put on a gene. And so this is another very useful aspect of using targeted nucleases to engineer cells. And as I mentioned, homologous recombination does happen by itself in cells when they try to repair double-strand breaks, but actually with the nuclease, we can increase homologous recombination tens to hundreds of fold, so this can be very effective in some cell types. And so now that we've talked about what nucleases do and why they're useful, the next thing is that not every targeted nuclease is going to be super effective at targeting a specific DNA sequence and introducing high rates of double-stranded breaks in a population of cells. And so one thing we like to do is we generate multiple nucleases for a specific project and then figure out which one of those nucleases has the highest gene editing activity or the highest ability to induce that double-stranded break. And so we have a number of assays at our disposal to actually quantify this induction of the double-stranded break or quantify gene editing. And so one of the first kind of assays that was developed as a universal method to do this was developed by a group at a company called Sangamo. And it's called the Surveyor Assay. It's also known as a T7E1 assay or the Cell1 assay. And so I'll just run you through the basics of this assay. Essentially, you transfect your target population of interest with your nuclease, and then you design PCR primers to actually amplify the region of interest that you're trying to induce double-strand breaks at. And what this assay is going to measure is the amount of indels, or the amount of mutations that occurred at that site in your population of cells. And so the trick to this assay is you take this PCR product, which will be made up of wild-type alleles, because some of the cells will not have indel formation, they will not have editing, and then you'll have some alleles that do have editing and they'll have insertions or deletions. And then what you do is you take this PCR product and you heat it up so that it becomes denatured, and then you slowly re-anneal it. And what will happen at some frequency is your wild-type alleles will anneal with mutant alleles, and you'll get a heteroduplex or a bubble forming where the DNA is not perfectly matched. And that's really where you get the readout of the assay, because once you have that pool of DNA strands where there is some heteroduplexes, you can actually treat that DNA in a tube with an enzyme called Cell1. And what that does is it identifies heteroduplex DNA and cleaves it. And then that cleaved DNA is what you actually quantify to look at the amount of nuclease activity. And you do this by simply running your PCR products out on a gel and using densitometry. And so here's some examples over here using zinc finger nucleases, where here you can see in the control where you don't use a nuclease, you get no heteroduplex formation and no cutting of your PCR product. But then when you do have indels and heteroduplexes, you see there is these smaller bands from those being cleaved, and then you can use densitometry and quantify that. And so if we were targeting a similar region with all of these, we would say this one is the most efficient nuclease. So this is the one we want to use for our project moving forward. So this was the first iteration of a universal method to quantify this. And actually, in the last 3 years or so, a new way to do this that's much easier, faster, and cheaper has been developed. And it's actually based on an algorithm called TIDE, which stands for Tracking of Indels by Decomposition. And essentially what these researchers did is they took fragments of DNA where they mimicked having indel mutations, and then they took Sanger sequencing results from those and did different iterations of this, and they developed an algorithm where you can literally take that same PCR product from this assay that we just talked about, and you can just send that for Sanger sequencing. And so normally if you do this with a wild-type PCR product, you get perfect Sanger sequencing. And if you have a mixed population where there's been gene editing occurring, there's going to be a bunch of different mutations at that double-stranded break site. And so essentially what you see is really good sequencing, and then right where you'd predict the cut site, the chromatogram becomes garbage. And that's because there's all those different indels, and so the Sanger sequencing can't differentiate what they all are. And so what these scientists did is they developed an algorithm where you can drop in the Sanger sequencing file of your population and a wild type as reference, and then it will actually tell you all the mutations that are in that population of cell, tell you what percentage of indels there are, what kind of indels there are. And so this is a really quick and efficient assay to figure out which CRISPR you're using or which guide RNA has the highest activity. And Brian will walk through exactly how to do all this at during his portion of the talk. But essentially, this is the readout from the assay. And so what it tells you is, on the bottom here is the size of the insertions or deletions in that population of cells, and then the frequency of those as well. And this assay is becoming very widely used and is actually comparable to what I'll talk about next, and that's analyzing your mutations by deep sequencing methodologies. And so this is another approach to analyze your indel frequency, typically not used for kind of day-to-day gene editing when you're just trying to make knockout cell lines. This is more frequently used when you're trying to do something for therapeutic uses, or you need really, really accurate numbers of the amount of gene editing that you're getting. And so this is done taking that same PCR product that we talked about earlier that's targeting your region of interest, and then simply applying Illumina deep sequencing to actually identify all of the different insertions and deletions in that population of cells and their frequency. And so for this, you can get a readout that looks something like this, where you have every read is indicative of a single allele in that population, and then you can use bioinformatics to actually quantify the frequency and abundance of each type of indel mutation in that population of cells. So that's what targeted nucleases are and how we quantify their activity. Now I just want to give you a little bit of background on the discovery and development of the CRISPR-Cas9 system. As like I said earlier, this is kind of the canonical system that we use for gene editing every day now. So interestingly, although you may not have heard of CRISPR before 2012, it was actually discovered incidentally in the 1980s by a group in Japan. Essentially, they were trying to sequence and identify genes in E. coli, and they found very interesting repeat sequences near one of the genes that they were studying in E. coli. And you can see those sequences shown here. And essentially what these turned out to be are the guide RNAs, or the tracer and cRNAs, of a CRISPR-Cas9 adaptive immune system in E. coli that they were studying. And so this was the first kind of even hint of the DNA sequences of the CRISPR-Cas9 system. Now, fast forward ahead decades to August 2012, and the actual discovery and idea of CRISPR-Cas9 being an adaptive immune system in bacteria was discovered by Jennifer Doudna and Emmanuel Charpentier. And so this was published in Science, and it basically just identified that it was a dual RNA-guided DNA endonuclease for adaptive bacterial immunity. And so essentially, this paper laid out lots of the foundational groundwork of what we know about the CRISPR-Cas9 system. So they figured out that in nature, there's actually a 2 RNA system. One is called the tracer RNA, and so now this is what makes up the backbone of the single RNA system that we use today. And then to target its specific invading bacterial phage in nature, you have a sequence-specific portion, which we call the protospacer. So this is what identifies the DNA. And then there's a region that's complementary to the tracer RNA, such that it makes a complete guide RNA. And so as you'll see in a second too, they also developed the single guide RNA system where they simply took this end and attached it to this and made it a single RNA rather than a dual RNA system. So they further went on to identify the nuclease domains of Cas9. So they identified the amino acids that they could mutate to make it a Nickase rather than a full nuclease. And they did all of this work in vitro in a tube. So they actually didn't do anything in mammalian cells in this manuscript. And then here's just the data showing where they actually made the chimeric guide RNAs, and they were even able to truncate the tracer RNA to make it smaller and still show that it has activity for cutting DNA plasmids in a tube. And so this was basically the foundation of CRISPR-Cas9 that everyone else kind of flocked to, and eventually, a few months later, was utilized for genome editing in mammalian cells by 2 groups at the same time, both from the Harvard MIT areas, namely Fang Zhang and George Church. And so they basically took the system that was described by Jennifer Doudna and Charpentier, and they adapted the system for use in mammalian cells. And so what they did is they took the Cas9, they humanized the codons, they put on nuclear localization sequences to make sure that went into the mammalian nucleus, and then they expressed the guide RNA, in this case just as the 2 separate RNAs that are found in nature. So this would be the CRNA that targets your gene of interest, and this would be the tracer RNA that is the backbone identified by the Cas9 protein. And then at the time, they were not sure if this accessory RNase 3 that was needed in other systems was actually necessary in mammalian cells. And it turns out that this is not required. In fact, there's RNases that exist in mammalian cells that actually carry out this task. And so that was very fortunate in helping with the ease of using this system in mammalian cells. And so the first thing they did is tested this 4-part system. And here, this is the Surveyor Assay that we talked about earlier. And so you can see they left out certain components. And when they had in the tracer RNA, the cRNA, and the Cas9, they could get detectable genome editing at their target gene. And then it didn't matter if they had that RNase that we just talked about. And so this was the first demonstration of gene editing in mammalian cells using CRISPR-Cas9. In that same paper, they actually did perform homologous recombination with the double-stranded breaks that were induced by CRISPR-Cas9. So in this case, they used a small piece of DNA to introduce a restriction enzyme site. And it's really hard to see here. This is an RFLP analysis, but there is detectable homologous recombination and integration of that HIN3 site, although it's at very low frequencies. This rate of HR with CRISPR has been vastly enhanced since this first publication. And so that's kind of the first demonstration of the use of CRISPR-Cas9 in mammalian cells. And so now one thing a lot of people talk about with gene editing is off-target activity. And so obviously when we design a guide RNA, we only want it to bind a specific site in the genome and introduce a double-stranded break only at that site. But of course it is possible that there are very similar sequences where your Cas9 might bind and introduce a double-stranded break. And so in the early days of CRISPR, there was a very heated debate over this, and I'm going to kind of walk you through how this all evolved and where it ended up and where we are today, knowing how specific CRISPR-Cas9 is for gene editing. And so shortly after those 2 science papers describing mammalian cell gene editing, there was a slew of off-target papers about CRISPR-Cas9 from very high-profile labs. And essentially, many of the papers claimed that CRISPR-Cas9 activity was not very specific in mammalian cells. So this is some data from just one of these papers where they looked at off-target editing of a specific guide RNA in mammalian cells. And just knowing a lot about CRISPR genome engineering, the first thing I can tell you, just by looking at this guide RNA, it is terrible. So basically, you can see how much homology it has throughout the genome. So these days, we would never use a guide that had this many sequences in the genome that were this similar to our guide RNA sequence. And so the data that they found is quantified here on the right. So this is the percent mutations or editing that they're seeing at all these sites. So you can see some off targets are just as high as your on target activity. And so this really got the field in an uproar about how CRISPR-Cas9 will never work for mammalian gene editing because it's not specific enough. And so what that ended up causing is a bunch of these labs trying to make CRISPR-Cas9 more specific by using different modalities of the system. But before I get right into that, like I mentioned, this is a terrible guide RNA. And so with lots of the learnings from people doing CRISPR-Cas9 editing, now we have very specific algorithms that can pick guide RNAs with super high efficiency. And one thing that's super misleading about some of these early papers is the specificity is actually in the first 13 nucleotides, and additionally that PAM sequence that I mentioned earlier. And so actually this part of the guide has very little to do with the sequence specificity of the CRISPR-Cas9 editing. But regardless, a lot of labs kind of jumped on this to publish very high-impact papers trying to make the system more specific using different modalities that I'll cover now. So the first one was a double nicking approach. This was published by Fang Zhang. This is where they actually mutated one of the nuclease domains of Cas9 such that instead of breaking both of the phosphodiester bonds of DNA, it only broke one. And the idea there is that is much less deleterious to a cell than a double-stranded break because that can be frequently simply repaired by the cell. But if you have 2 Cas9 molecules juxtaposed to each other, then essentially you get additive specificity. So if this guy is off target somewhere else, makes a nick, that was likely to just get repaired. But when you have them in this kind of proximity, what you're really doing is making a double-stranded break. And so what they showed is that you could actually get high rates of gene editing using this approach. So here, just the quantification of indels at 3 different target sites, anywhere from 10 to 40, 50% gene editing. And so this approach, at the time, everyone was pretty excited about it. But of course, it adds a lot more work as you have to design 2 different guide RNAs. They both have to function, and then you hope to get high rates of gene editing. And so here's just some more data demonstrating that the off-targets basically go away with this system. So here's the single Cas9 nuclease, and then you can't even see the bars for the off-targets with the Nickase approach because there are no off-targets. So after that, Kees Jung actually took an approach very similar to Talon's, where instead of using a Nickase, he actually used a dead Cas9, so both domains were mutated, so simply it could just bind DNA. And then they took an approach very similar to the Talons, or the zinc finger nucleases I told you about earlier, where they actually fused a FOC1 nuclease to the Cas9 protein, and essentially they worked just like ZFNs or Talons, where you would have 2 of them juxtaposed to each other. The FOC1s would dimerize and induce a double-stranded break. And so they developed different methods of either having the guide RNAs in an orientation so the PAMs were into each other, or so that the PAMs were out from each other, and they found that the PAM out configuration worked best for inducing double-stranded breaks with this approach. They showed that it could work at many target sites in the genome with good efficiencies, quantified over here for a bunch of different genes. And then they showed again, just like the Nickase approach, that all the off-targets go away. So that's all well and good. Again, it's more work. You have to design multiple guide RNAs, and they both have to work for you to get high rates of editing. And so then very recently, what people did is they actually did some protein engineering of the Cas9 protein to actually inherently enhance its specificity for genome engineering. And largely, these studies focused on mutating amino acids in the Cas9 protein that essentially made non-specific interactions with DNA. And so by mutating specific amino acids in the protein, they were able to make these high-fidelity versions of Cas9, as they called them. And so this is a lot to look at, but essentially this is a deep sequencing method to look at off-targets. And with the wild-type Cas9, you can see all of these off-target sites for these different genes. And with their hi-fi Cas9, they essentially all go away. So they claim that this Cas9 has a higher specificity, and they also claim that it has the same on-target efficacy as the original Cas9. So shown here is just gene editing percentages at a few different genes using the wild-type Cas9 or this hi-fi Cas9 that they developed through protein engineering. So I will just say all of this has been very interesting to watch in the field, but the reality of it is that with the algorithms that we use today, CRISPR-Cas9 can be extremely, extremely specific. And for research purposes, I would say that there's no need to even use a hi-fi Cas9. My laboratory, we've done a lot of different analyses looking at off-targets of guide RNAs that have been well designed with the most recent algorithms, and we've developed a number of guide RNAs that have absolutely zero off-targets in the genome, testing them in many different cell lines. So I don't think there's anything wrong with using these hi-fi mutants. Sometimes they're sold to be a little bit better than they are, i.e. the on-target activity is not actually as good as the wild-type Cas9. And like I said, if you use the algorithms that Brian will cover today to design your guide RNAs using the wild-type Cas9 for research purposes is, I think, absolutely fine. And so on that note, I'm going to transition into some of the other uses of the Cas9 protein that some of you may not be aware of, but may be very useful for your research. And so a lot of this is based on using the dead Cas9, so the nuclease-free Cas9, essentially using it as an ability to bind any sequence in the genome and then bring other molecules there to do specific functions. And so if you go to the website, AdGene, they are actually a plasmid repository, and you can look at all of the different modalities of the uses of dead Cas9 and regular Cas9, so it gives a really good overview. So you can use it for double-strand breaks, nicking. You can actually use it to suppress transcription of specific genes, gene activation. You can actually use it to bind specific pieces of DNA and pull them down and see which proteins are bound to them. You can label DNA with fluorophores at specific sequences by fusing Cas9 to things like GFP. You can do screens in bacteria, and then you can also tag genes. And so I'll go over a couple of these different systems just to give you a flavor of all the abilities of the CRISPR system for doing things other than introducing a double-stranded break. And so this is just, again, a reiteration of what we typically think of nucleases doing, but what actually people have done here for gene regulation is using the dead Cas9 to either activate genes or hyperactivate them transcriptionally or repress them transcriptionally to silence them. And so the very first systems, they actually used the guide RNA, and they engineered it to have an extra portion that actually stuck out of the Cas9 protein. And they encoded in this a sequence that's recognized by a specific protein called MS2, and they fuse this MS2 to different domains that would help with gene activation or gene repression. And so for gene activation, the first systems used a VP64, and so what this does is it recruits transcriptional activators and helps overexpress your gene of interest. And then to repress expression, they fuse it to a CRAB domain, which can actually help repress transcription of a specific gene of interest. And so these first systems worked okay, but actually in the last few years, a very robust system for gene activation was developed by Fang Zhang's group. In this work, they actually used 2 MS2 loops in the guide RNA, and what they did is they fused that MS2 protein to a protein called P65 and HSF1. And then they also kept the VP64 element, and what this essentially made was a super hyperactivation system, so you can turn up gene transcription something like 1,000 to 10,000 fold on average. And they showed this at many, many genes, and they actually even did genome-wide screens using this system. And so here's just an example of a bunch of different genes where even where they would target the old system, the VP64, with many guides at once, versus this new hyper system in blue. You can kind of focus on that. You can see the fold activation they're getting, like 10, sometimes even 100-fold activation of that gene. And so this can be really useful for overexpressing endogenous genes. And one other caveat of this is that there's also design rules for this system whereby you target kind of the upstream region of the transcriptional start. And as you can see here, some of these, each one of these is an individual guide RNA that they're testing for gene activation at these 4 different genes. And you can see that some guides don't really work well. And so there are design rules and algorithms out there that will help you find guides like this where you'll get really robust gene activation. So conversely to gene activation is gene repression, and as I mentioned, for this they use a Crab fusion. So originally, actually, people used just the dead Cas9 and tried to park that on the 5' UTR of genes, and they found that that physical binding of Cas9, because it's such a large protein, that in itself could actually repress transcription. And so you'll see that here in their control where they just use dead Cas9. However, when they fuse it to this Crab domain, it actually enhances gene repression. So this is just 2 examples at 2 different genes where they're testing different guide RNAs to repress the expression of these genes. And you can see that even Cas9 alone can repress gene expression, but the Crab fusion helps. But the punchline of this is the system works okay. It's really hard to get 100% gene repression with this system. Sometimes you can get 50 to 80%, which may be enough for your system in identifying a phenotype of interest. But I'm certain there's a lot of work going on in this field by many people to develop systems that can actually completely silence gene transcription using a dead Cas9 fusion of some sort. So as I mentioned earlier, you can also label specific chromosomal sequences for live cell imaging using Cas9 fused to GFP. So here's just some data from one paper doing this, where they actually targeted regions of the genome, and they used multiple guide RNAs to enhance the signal of that GFP so they could quantify those regions on human cells. And so interestingly, they actually compared this directly with fish. So they took cells and performed a fish analysis, or they used this CRISPR-Cas9, and the CRISPR's in green, and then the fish is in red. And what you can see is that the overlap is quite good with fish, indicating that these are likely very specific events, making this a useful system. And so they go on to do this first at a couple genes where there's repetitive regions, so they can actually get a lot of guide RNAs binding near each other to get a signal to noise ratio that's good enough to see these punctate spots in cells where these genes are located. And in this case, I think there's 3 copies of the MUC gene that they're looking at. And then what they figured out is the number of guides you had to have in a small region of DNA to actually get that signal high enough to see punctate spots in a cell. And essentially with this first iteration of the system, what they found is they need about 16 to 26 guide RNAs in a single area to actually get the signal high enough to see these locations. But there's actually been a couple papers in the last year or so where they've actually amplified the system in different ways using fewer guide RNAs. And they've actually been able to label 2 different chromosomal regions with different colors and actually see them come together as the DNA interacts to look at DNA interaction in living live cells. And so there's lots of really interesting uses of this technology as well. So the last technology that I'm going to talk about, because it's really interesting to me, and I think it's really cool, is the single base editing Cas9. So this is a technology that was developed by David Liu at Harvard. And essentially what he did is he first took a dead Cas9 and he fused it to an enzyme called ApoBec. You may know of Ruben Harris here at the U studies this extensively. And essentially what this enzyme does is it's a deaminase. And so it will actually mutate C's to U's in an RNA sequence usually, but actually they found it also works on DNA when it's in a single-stranded form because it's bound by Cas9. And so essentially they took this dead Cas9, fused to ApoBec, and in a tube they tested its binding of a target site. And so here is the 20 base pair target site that they were looking at essentially. And what they found is it actually opens up a window of editing between bases 4 and 8 within your target site, and it will actually convert any C's in that window. In this example, these are all C's, any of those C's to U's. And so this is just an in vitro assay to look at the ability of this system to convert C's to U's in a tube. And so what they did with this is the hope is that once you make a C to a U in a cell, that it possibly could be repaired by a specific pathway to turn that C into a T. And basically this gives us what we call a base editor technology where we can site-specifically change any C into a T in the genome. And so the logic behind this is actually, again, relying on the way a cell repairs the mutation that you're making. And so shown here is a C in the genome that they wanted to change to a T. And they basically had to develop different iterations of the base editor, ending up with this base editor 3 that actually functioned very well in the cell. And I'll kind of walk you through how this works. So basically, if you change a C to a U in the cell, it's typically going to be repaired by cellular uracil DNA glycosylase. And that's going to make an A basic site, and then it's going to use base excision repair, and essentially it gets converted right back to a C. You know, this is what we want to happen in our cells. We want them to be repaired perfectly most of the time. And so the first thing that they did is they fused the Cas9 to a UGI. This is a protein that actually it's an inhibitor of uracil DNA glycosylase. So they're blocking this, essentially blocking it from being repaired perfectly. And then to further push this system to work at high efficiencies, they actually, instead of using a dead Cas9, they used a Nickase version. And the reason for this is that during cell division, if a cell comes upon a UG mismatch, it actually uses a specific mechanism whereby they look at the nicking. So cells use a nick strand to basically determine which one's the parental strand to preferentially use during DNA replication. And so by doing this, you can actually force the cell into thinking that this is the parental strand, so you should fix it this way. And essentially what that leads to is changing your C to a T at a site in the genome using this base editor technology. And so here's just some results at different Cs within target sites in many genes in the genome. And what you can see here in green, this is the base editor 3 technology. You can get anywhere from 30 to 50% conversion of those alleles in a population of cells. And if you try and do this with homologous recombination and Cas9, it's in blue, and you can't see those because they're around 1%. And then the previous iterations I mentioned, without the Nickase and without the inhibitor, you still get a little bit of base editing, but not as high a frequency. And so this is the type of results that you can get with this base editor to change specific Cs to Ts. And so by going to some conferences recently too, David Liu has not published yet, but he has talked about the fact that he's made other base editors. And so he's made base editors with the ability to change specific C's to G's and also A's to G's. And so in the end, we believe there will be a tool set of base editors to change any base in the genome to any other base in a site-specific manner. And so that you can imagine this can be a very powerful technology for manipulating the genome, studying different SNPs, or changing amino acids with high efficiency. And so the last thing before I talk about other CRISPR systems is just a little touch on the CRISPR patent battle, just because you see this in the news all the time and it's kind of an interesting topic. So just to give you an idea of the complexity of what's going on with the patent landscape, you can see that all of these different people that are involved with this and who has foundational patents and secondary patents, and actually even the university and a spin-out company, Calyx, they own a little bit of IP, or they think they do, of the Cas9 system. And so it's really interesting to watch this all unfold over the last 5 years. So recently in the United States, the Broad and MIT, they were granted the license for CRISPR-Cas9, and then about 2 weeks later, all of Europe granted the patent to Jennifer Doudna and Emmanuel Charpentier. And there's been lots of kind of battling going back and forth. And I think the punchline of this is we won't know for many years who really owns CRISPR-Cas9. There's even been talk of kind of making a pooled patent landscape, kind of what they did for all the different the music formats back in the 90s when there was MP3, MP4. What they did is they all came together with their patents and they made one entity that licensed out patents. So essentially they all got a little bit of the pie. And so some people think that's what's going to happen with CRISPR-Cas9 as well. And then the last thing I'll touch on before Brian comes up, it's kind of interesting because there's all this talk about patent battles and usage and things with CRISPR-Cas9, when the reality of it is that in nature there's hundreds and hundreds of other CRISPR systems besides Cas9, and they're being discovered daily. And so in the end, we're going to have this giant toolbox of different nucleases to utilize besides Cas9. So just to highlight one of these, recently discovered in the last couple years by Fang Zhang's group, all through bioinformatic mining of databases that are publicly available, they found this family called CRISPR-CPF1. So this is distinct from CRISPR-Cas9, but has a very similar function in bacteria where it's part of the adaptive immune system. There are some very subtle differences between the systems. It seems that there are more accessory proteins required for CRISPR-Cas9. So they have these other proteins that help process and capture the guide RNA sequences, and perhaps CPF1 has less of these. And so what they did is they figured out how to get this system to work in human cells. It functions very similar to Cas9. It has a slightly different requirement for its PAM sequence. It's actually a T-rich PAM. It actually has a little bit longer guide RNA sequence. And you can see here in the Surveyor data over here that it has very good activity in mammalian cells, even targeting the same genes as Cas9. So this is CPF1 from 2 different bacterial strains. You can see that you can get very good rates of gene editing with this system as well. So that just gives you the idea that there's going to be hundreds of different systems available over the next decades that can be used for genome engineering besides CRISPR-Cas9. And some of these may end up being much better than CRISPR-Cas9, perhaps having slightly better on-target activity or higher nuclease activity, or they're just generally better for genome engineering for various reasons. And so with that background, I'm going to hand it off to Brian, who's going to get into the nitty-gritty nuts and bolts of how you actually design CRISPR-Cas9 guide RNAs such that you have high on-target activity and low off-target activity, and then also how to actually implement these in cultured mammalian cells, how to isolate single cells and analyze those cells to determine if your cell has gene editing and if it is potentially knocked out for your gene of interest. So with that, Brian.
Thank you, Branden. So I'm going to be going over the steps that we take to generate your knockout cell line. We do this in the Genome Engineering Shared Resource. We've been doing this for now 2 years, and even before that I was working with CRISPR-Cas. So the first thing you want to do when developing or designing your project to knock out your gene of interest is you want to identify the genomic region to be targeted. The main thing you want to look at first is make sure that the exon present is present in all transcript variants, and you can use the UCSC genome browser for this. The website's shown here, and I'll be going over that briefly in the next few minutes. You also then want to pick an exon that's upstream of, or part of an important functional domain, and you can use interpro or motif for this. And also then another important point is that you want to find all the possible protospace or guide RNA sequences around the intended target site. And there's been many labs have designed online resources for this, shown here, but I'm going to be focusing on a couple of new websites that have become available recently, CRISPOR and guides. So the CRISPR web page, this is the front page of it when you go to the CRISPR website here. And it's basically just 3 steps. Step 1, you enter your the region of interest that you want to target, an exon or a region around an exon. You then select the genome that exon is within, and then you can choose the protospace or adjacent motifs that you're going to be using, the specific Cas9 that you're using. And the cool thing about this site is they've integrated all the most recent Cas9s. They're constantly updating this. This website became available in 2016, and it's been updated as recently as April of this year. But I imagine that they'll be adding more to this list in the future. The other website that I mentioned was guides. And guides can be used it's been designed for doing custom guide RNA libraries, but for this, the purposes of this talk, I'm talking bringing you can use it for creating or picking guides for a specific gene. So again, you just enter the gene name and the number of guides you want for that and the genome that you'll be targeting, and it'll give you a readout similar to this, where you actually have all the exons of the gene of interest, and it integrates RNA-seq data showing expression levels, and if you click on either of these or toggle over, they'll show you the expression levels within specific tissues. And importantly, there's also functional domain data that this one provides. So again, just a reminder, find all the protospace or guide RNAs around your intended target site. And then finally, you want to select at least, as Branden mentioned earlier, you want to select multiple protospace or guide RNA sequences to ensure that you get efficient cutting by at least one of them. So the UCSC genome browser is where I usually go to first when I'm designing a project. This is the front page of the UCSC genome browser. And if you click on the Genomes tab here, you'll be brought to a page that looks like this. And here you can then pick the species that you can be targeting, and again, enter the gene name in that window and press go. This will then bring you to a page where you'll have your known genes, the gene of interest that you're looking at, plus any isoforms or any other related genes to that gene. But what I want you to focus on here is really at the bottom of the page, the ref-seq. You want to click on get the reference sequence to that gene, and this will bring you to a window that looks like this, where you have the genome assembly along with the chromosome and the location on that chromosome where your gene resides. And highlighted will be here. It's really small, but this is the part of the site you want to click on for to get the ref-seq. This will then bring you to a page that has your gene name, a lot of other links that you can use for your project, as well as a summary of the gene and what it does. And at the bottom will be the messenger RNA genome alignments tab. So you click on that, and that eventually gets you to where the nuts and bolts of the whole thing. So conveniently, they've highlighted the 5' and 3' untranslated of that gene, the cDNA, as well as all the coding sequences in blue with the splice junctions from each of the exons in light blue. Further down on that same page, you'll find the genomic locus, where they've included the introns spanning your exons of interest. So in this case, I'm just using exon 6 of HPRT as an example. You then select and copy at most 1,000 base pairs, and it's for the CRISPR website, and enter that into the window pick again your genome, your PAM, and click that, and then you'll end up getting a page. It'll go through and it'll find all the guides within the entire 1,000 base pairs. It'll categorize them as either green, yellow, or red. Green being the best, red being the worst. Green is any one with a score of 50 or greater. Yellow is anything 21 to 50, and then red is below that. And what you want to for this purpose, what we're really trying to do is target the exon of interest. So this portion here, you can actually slide. It's a sliding window. You can slide over to the portion of that 1,000 base pairs, which ends up being about 460 to about 545 is where the exon is. And you focus on trying to find, these are the guides that you would want to use to target this specific exon, because you want to have the indels within this exon to put the protein out of frame. So below that same tab is a table that has all the information, the position, the guide sequence, the specificity score, the predicted efficiency, and other information here. I've taken a screenshot of that window, but I've focused farther down on the list. There were guides on that list that were higher, but the highest guide that was within the exon is this guide here at position 537. And so this is the one I am going to be using for the demonstration of this in this talk. So the cool thing about this, about CRISPR, in addition to finding all the guides and having all this other information, they also integrate the restriction enzyme site information, so that if you're doing your editing or on your cell line and you want to find out whether or not you have edited clones, you can use a restriction enzyme to digest the PCR product and look for the absence of cutting if there's been an edited event. And unfortunately, this one that I'm having the example doesn't have that, but you can choose ones that have ones with the restriction enzyme. But they also have cloning and PCR primer data here. So if it's a quick click to that, and you'll end up having go to another page that has, again, your guide sequence, and below that, in table of contents, these, again, are clickable that will then bring you to the lower part of the page. They'll have information for cloning your guide RNA into any system that you want. In the Genomic Engineering Shared Resource, we predominantly use U6 expression vectors provided by AdGene. And it also provides information to our PCR primers to amplify your target sites, as well as if you're interested in off-target sites, you can do that. So this is the PCR primers that are available for that one. So the site will generate or find PCR primers that span your site, and usually set a cutoff of about 400 base pairs. And then this, for cloning your guide RNA into the U6 promoter driven plasmid, there's multiple plasmids that can be picked from with this tab as well. This is just one that is used for the Zhang Labs BBS1 golden gating system. So again, just a reminder, this was the exon that we were targeting. This in black is intron, green is the coding sequence, and this is the guide RNA and the PAM sequence that were available for that. So to clone these, we've used a golden gating system where we, as I mentioned before, there's BBS1 restriction enzyme site overhangs on the oligos that you design that have the 20 base pairs. So you have a sense oligo with a 4 base pair overhang and an antisense with another 4 base pair overhang. You put them in with your plasmid that you're cloning them into, and you would first anneal them and then perform a BBS1 restriction digest on the plasmid and a vector ligation. And it's a really efficient and cost-effective way of making an expression vector. And so once you have the vector, you'll want to target your cells. So we would just go through the targeting workflow. You transfect the cells with your CRISPR expression vector by the means that you want to. Wait several days, enrich the target population. You can do flow cytometry. We have Cas9s that have GFP co-expressed with the Cas9, so you can do flow cytometry for the target cells. Or if you don't want to do flow cytometry, you can certainly do transient antibiotic treatment on them to kill off the non-targeted cells. You then expand those cells over for a few more days to get enough cells in the pooled population to do the tide analysis, and I'll get into that in a little bit, but I'll continue on with the workflow first. Once you have your tide analysis done, you can subclone those into either 96-fold plates or other methods to get a single colonies from those. And then again, want to do this only after you've confirmed that your CRISPR is actually cutting. You don't want to clone your cells when if the CRISPR is not cutting efficiently, you don't want to really go through this step. This then is another few weeks to get to the point where you'll end up expanding those clones. You can either replicate them onto 96-fold plates for direct lysis and analyzing them that way, or sometimes we expand them into 24-fold plate formats and do genomic DNA preps from those. And then finally, you want to do sequence analysis on them, collect the genomic DNA from the individual clones, PCR amplify across the region, and sequence the resulting amplicons. And you can, again, even for these, at this point, you can either you can do tide analysis as well on these, and I'll show you examples of tide analysis on individual clones. And then if necessary, you can topo-TA clone these amplicons to segregate the alleles. So tide analysis, again, as Branden illustrated earlier, you have a wild-type trace file, and this is the protospacer guide RNA sequence for this particular trace file in the PAM site shown here in red. And the CRISPR is going to cut here 3 base pairs upstream of the PAM site. And you can see that in the targeted population, the sequence mostly goes haywire immediately downstream of that. There's actually, you can see some editing even upstream of that. And that's indicative of deletions that are going 5' to the cut site. This is the sequence alignment window. And so you have perfect sequence alignment here in the first few hundred base pairs. And right before that, you can actually see a little bit of a shoulder where the sequence is starting to go, starting to align, and then it's really not aligned at that point. And the readout again here is where you have 60% targeting efficiency with this particular guide. This was a really efficient one. It's only about 10% of wild type is still within that population. And so a few tips and tricks about tide analysis. So there's advanced settings in the tide page, including the alignment window. It's usually set at 100 base pairs. The left boundary is 100 base pairs from the start of the sequence, because the sequence is usually not good in the first 100 base pairs of any sequence read that you're doing. The decomposition window should span the entire sequence of your amplicon. Oftentimes in Sanger sequencing, there are times when if you're sequencing a short PCR product, the sequencing will continue to sequence even after it's gone past that, and so you want to try to keep this window up to what you want. And then finally, the indel size range. You can adjust this to different lengths to get to find sequences or indels that are bigger than 10 base pairs. So you want to ensure that you have an accurate trace file for this. The break site should be located about 200 base pairs from the sequence start site. And so you might have to sequence from either the 5' or 3' end to get that to work. You also then want to adjust the decomposition window to span only the amplicon's true size. And an R-squared value of 0.9 or better is desired. And then finally, for some sequences, as I mentioned earlier, increasing the size range can reveal large deletions or insertions. But the one thing this can't do is it can't discern single nucleotide substitutions within your population of cells. So that has to be done by straight sequencing. That might actually come up during when you actually look at when you compare your trace file to your sequence file to your reference sequence. And so this is, once you have your individual clones, and you do tide analysis on them, what you're trying to get is a biallelic knockout. And so what you can see here is we have, this one is a -1 and a -2 by tide, and a score of these are all scores of 0.98, so that would be one clone that you'd want to use. Homozygous knockout of -1. This one is a biallelic with a +1 or a -5, and then finally you wouldn't really want to, if you want a heterozygous clone, you can keep these for other purposes, but for straight knockout purposes, you wouldn't want to use one that's wild type over a -1. And so then the GESR offers many services for making any of your reagents or doing cell lines for your projects. This includes non-phase billing, including we have consulting hours, consulting rates, and then guide RNA design service, expression vector construction, guide RNA validation, and Cas9 expression vector reagents. We also have phase billing, which includes the bigger projects like making the cell line. This includes, I just demonstrated the single knockout of a non-essential gene, conditionally null alleles for essential genes, single amino acid knock-in mutations, and also some Cas9 whole genome. We haven't done these yet. We are in the process of getting ready to do some of these in the future, but those are project specific. And finally, we have now, in the last year, we've started coordinating with the MGL to make mutant mice by CRISPR-Cas. And then the Genome Engineering Shared Resource is now celebrating its second year in existence. We started last year in fall of 2015, or 2 years, 2015. And these are the projects that we've been able to complete so far in that time. We've had many guide RNA design services and expression vector constructions, quite a few single knockout of non-essential genes, as well as conditionally null alleles and some knock-in mutations. And just recently, a few months ago, we were able to make 2 gene knockout mice by pronuclear injection of RNPs. And with that, I'll take any questions. Yeah.
So Branden actually spoke using CRISPR-Cas9 to make labeled proteins in vivo. Could you just expand a little bit on the strategy to do that?
Using it to label protein? Oh, like tagging protein?
Yes.
Yeah, so if you want to tag an endogenous gene, you can use homologous recombination to put a small tag on the last exon or the first exon of a gene. You could tag it with GFP or with a MIC tag or a flag tag, so you could actually pull that down. And so that's something that's pretty trivial for us to do.
Has anyone done this to look for proteins binding RNA? Suppose you're looking for RNA regulation with a Cas9. Since it's single-stranded, it's possible to look at that.
So with Cas9, it can't really bind single-stranded RNA. There is one obscure way to make it work, but it doesn't work very well. But there's a new system of CRISPR called C2C2 or Cas13a. This is a single-stranded RNA binding protein that works just like Cas9. It has a guide RNA, but instead of binding DNA, it binds single-stranded RNA. And so those systems are becoming available. There's some nuances with them, but newer versions are being found again from bioinformatic mining of our databases. So I think that's something that could be done in the near future.
For time analysis, is the APCON true size of what's that mean? just get the PCR fragment side when we set up the decomposition window. Okay, another question is that's a guard RNA. So that means when we synthesize the fragment sequence, we need to use the U replace the T, or we just use the T?
I didn't get the second question.
Yeah.
I mean, that's called guard RNA, right?
Guide RNA, yep.
Right. It's a DNA sequence that you order.
Yeah.
Like yeah.
If you go back like to the plasmid where you clone in your guide RNA sequence, so this U6 promoter, it's a pull-3 promoter that expresses transcripts in mammalian cells, and so that will take your DNA plasmid and make an RNA transcript off of it in the cell, and that will be your guide RNA in the cell. And then you had a question about the true amplicon size and those
yes,
settings.
Kind of analysis, when we set up the decomposition windows.
Yeah, so what he means is like if you have a 300 base pair fragment and you send it for Sanger sequencing with a forward primer, sometimes it'll come back with 800 base pairs of sequence. And that's because it basically, the algorithms they use to analyze your data are not sophisticated enough, especially when it goes into a bunch of messy peaks. And so what he's saying is just set that to the true 300 base pairs starting at the beginning to the 300 base pairs. Otherwise, Tide has problems figuring out the indels.
Yeah. I've been able to even find a 34. So at one point I was doing a project where I was inserting a LOX-P site, a 34 base per LOX-P site, and I was able to use Tide to show that in the population there were clones that incorporated that 34 base per LOX-P site by expanding the amplicon size window as well.
So can we transfect the guide RNA as an oligo directly without
Yeah. So basically, the way that we described using the CRISPR-Cas9 system here is basically on the cheap. So this is using all plasmid-based systems that are very affordable, like we showed. You can make a guide RNA for $20. But in fact, you can now order RNA oligos of 100 base pairs that encode the entire guide RNA, and you can deliver that to a cell as a guide RNA for gene editing. The ones that are currently available to use with protein are pretty cheap. You can get them for maybe $150 if you get the 2-component RNA. So if you order the tracer RNA and the CRNA for your gene and use that with Cas9 protein, you can get them for around $150. So those are called RNPs, ribonuclear protein complex Cas9. And there are these really advanced reagents that my lab uses, especially for primary cells, where you can actually get that 100 base pair RNA and then make chemical modifications on the end of it to make it super stable, and you can get like insanely high rates of gene editing with those, but those are like about $1,000 a piece, so they're really expensive. So it is possible.
How well does this work for the mitochondrial genome?
That's a great question. So to date, there's only been one paper on CRISPR-Cas9 being used to edit the mitochondrial genome. And actually, I kind of know a lot about this because I'm part of a startup company in town that actually focuses partly on mitochondrial gene editing. And this is a huge problem in the research field because there is no effective way today to gene edit the mitochondrial genome. And one of the reasons for that is the mechanism by which the mitochondrial genomes are repaired is essentially a black box. No one really understands how they repair the DNA in the mitochondria. And so that no one has yet been able to manipulate that using genome engineering. But there is some stuff coming out of Mayo, specifically Steve Ecker's lab, where they've developed some methods to actually make deletions in the mitochondrial genome, but it's still very complicated and definitely not optimized. All right, so if you have more questions, I think this PowerPoint will be put on the website, so if we went through too fast, you should be able to get that. And otherwise, if you have any further questions about genome editing, like Brian said, we have consulting hours, And also, if you have any projects that you need help with, we would love to help you with those. And if you have any specific questions or quick questions, feel free to email Brian or myself, and we'll try to get back to you as soon as possible. So thanks for your attention.